Alrighty, we're going to make a start, and any other groups can sort of join in as they arrive. So, good afternoon, everyone. My name is Em, and I work at the Welcome Genome campus. And today's session is our second in our Genomics Lite, the Human Genome Project series, where we're going to be discussing lab techniques. With us today, we have Leah Chappell, who'll be discussing some of the newer lab techniques that she uses in the lab. And we've also got Fran Gale, part of the public engagement team with myself, who'll be here to help answer any questions or technical issues that we hopefully won't have. So a very brief introduction then to the campus. So this is the campus itself, located just outside of Cambridge, which is where we would usually be if it wasn't for this year. And on the campus, we have two main scientific research institutes, the Wellcome Sanger Institute and EMBL EBI, which is mainly sort of biodata focused. I've also got a really quick video to show you, just to give you a bit of context to the campus for those of you who might not have heard of us or visited before.
Greenwich, London, site of 0 degrees longitude. Geneva, Switzerland, home city of the CERN Large Hadron Collider. Just as the Greenwich Observatory is considered the home of time, and CERN the heart of nuclear physics, so this campus on the edge of Cambridge is forever linked with the science of genomics. The Wellcome Genome Campus is where the first animal genome of a simple nematode worm was sequenced in the late 1990s. And now the campus is leading a revolution in genomics and biodata, bringing the prospect of previously unimagined medical breakthroughs. So, scientists from all over the world are attracted to work here. Let's see how they define what a genome is.
I would say the genome is a blueprint of all the information needed to generate a whole organism.
Every organism has its own genome. Yeah. Just as the genome connects us with all other humans, you know, we all share much more of our code than we have that is different.
Not only between each species is the genome different, but actually each individual of the species has a slightly different genome.
It's those instructions that define how the natural world looks.
That is much better than I can explain.
Awesome. So hopefully that gave you a nice bit of context to what we get up to on campus. And so we have sort of two halves to this session. Firstly, we've got a video from our colleague Kim Judge, who wasn't available to come along today, but has filmed using some really new sequencing techniques at home, so we can see how that is used. And then Leah, who's joined us today, will be talking about different aspects of newer sequencing techniques, and we'll be able to answer some questions towards the end of our session. If you've got questions, pop them in the Q&A box as we go, so that we can get some answers for those as well. So I'm going to stop sharing that, and I'm going to find our video from Kim. So hopefully you can all see that. Fran, is that okay? Fab.
Hi everyone, thank you for joining me here today for this video on using sequencing technology. So my name is Dr. Kim Judge, and I work at the Sanger Institute, and I specialise in long-read sequencing technologies, and one example of that is this machine. So this is called a MinION. It's one of the smallest sequencers that's available. You can see how it fits in the palm of my hand. And when we open the lid up, we've got this flow cell inside. And we can just plug this directly into the laptop that I'm filming this on and do some DNA sequencing. But before we can start sequencing, we have to prepare our DNA. So that's what we're going to do first. So I've got some DNA here in this tiny little test tube. We call this an Eppendorf tube. And the first thing I'm going to do is add it to some chemicals that are going to prepare it for sequencing. So I'm just opening them now. we're using a special version of these chemicals that's called a field sequencing kit. So this is to enable us to take this sequencer out out the laboratory and go anywhere with it, and we can do all kinds of different science with this. So that might be ecology, it might be remote medicine. This MinION has been used on the space station as well, so that's very exciting. Okay, so the first thing I'm going to do, I have this little tiny rack of chemicals here. I'm going to go into the first one with my DNA. I'm going to use this pipette and just take my DNA out the tube. Now what we're not doing here is extracting the DNA from cells and purifying it, so that's already been done. If you want to know a bit more about how to do that, I know the team at Sanger have got some great videos including how to get DNA out of a strawberry, so check that out. We're going to put that into here like this, and we're going to then squirt it up and down to give it a really good old mix, because we want to get everything nicely combined together. So what I've got here in this tube is something called a transposome. It's like a little protein machine, and what it's going to do is it's going to jump into the DNA and cut it and put some predictable parts of sequences of DNA either side of that cut, and we can use that to help us get started with the sequencing. So now I've got my DNA in there, I have to incubate it at 30 degrees for 1 minute. So in a lab we would use some equipment called a thermal cycler to do this, but here, because I'm not in a lab, I'm just going to use the warmth of my hand to do it. I've usually got really cold hands, but it does help wearing gloves. So we're going to keep it nice and warm. And the reason that we're incubating it at about 30 degrees is because we borrow these machines from nature, and as a lot of proteins in nature are adapted to work well at sort of optimal cell operating temperatures, so kind of in the region of 30 to 38 degrees. This is the temperature that this molecule is going to operate really effectively at. Okay, so just a few more seconds. So again, if we were in a lab, we would have a machine that did this automatically for us. But what we need to do now is heat it up to about 80 degrees, and that's going to denature or kill off or just break down that machinery that's inserting into the DNA and stop it from continuing to chop up all the time. Here I've just boiled my kettle, and I'm going to put it into this really hot cup of water. You can't see it steaming, but it is really hot. So I'm just going to go in there with that. And again, we're going to leave that for about a minute, and that's going to be long enough to inactivate that machinery. It doesn't have to be a red spotty mug. This is just the one I have. So this is a really simplified method of preparing DNA. It's been developed by the company that makes the MinION for making it straightforward to prepare DNA out in the field. When we are at the Wellcome Sanger Institute, normally we have some slightly more elaborate methods. They can involve a lot of lab equipment and robots and things like that. They are really efficient and really good at preparing the DNA as optimally as possible. So we make sure that we wring every last bit of DNA out of our sample and sequence it really well. This method's a little bit less efficient, but it is very straightforward to do in a non-lab based setting. Okay, so just a few more seconds here in activating. Okay, and that is the time up. So what I'm going to do next is I'm going to take the prepared DNA and I'm going to go into my next tube. I just have to find what I did with it. Okay, so we're going to go into that second tube with our prepared DNA. And again, we're just mixing it all up and down. Okay, now we have to leave this to incubate for 5 minutes at room temperature. So that's a little bit of a long time to wait. So I'm just going to see you in 5 minutes. Okay, so the last thing we need to do is make some buffer, which is going to be the solution that we use to load our DNA onto the flow cell. So to do this, I'm going to use a slightly bigger pipette. And the great thing about these pipettes is we can get them to measure very precise amounts of liquid. So I want to use here 65 microliters. So that's 0.065 of a milliliter. So if I take this and turn it until it says 6.5 on it, it's kind of hard to tell because to me this is back to front. There we go. Okay, and I'm going to put one of these plastic tips on it. So we use these so that we don't contaminate the liquids that we're using. And I'm going to draw up 65 microliters. So I do that by just pushing my thumb down and then lifting my thumb up. And that will suck a little bit of liquid into the tip of my pipette there. Okay. And then I'm just going to add it to the third tube here, just while my DNA finishes incubating. Okay, so in that goes. We need to make sure it's very well mixed again. There's quite a lot of powder that we need to resuspend this time, so giving it a really good mix there. Okay, so that's looking good. Alright, so we're about ready to load up our flow cell. Okay, so my next step is going to be to prepare the flow cell for the DNA coming onto it. So the first thing that I'm going to want to do is just mix up this solution. And then I'm just adjusting my pipette so that the number is much higher because I want to take up a greater volume of liquid here. Okay, so that's about right. So we're going to just take some of this solution here. That's well mixed. So what I've got here inside my sequencing machine is this small flow cell, and that's where my DNA is going to go. So the first thing I want to do is peel back this sticky tape, and that's going to expose a little hole, whoop, just there, and that's where I can pipette into. So making sure I've got no air in the tip of it, so we don't want air to get in, just liquid. And I'm going to just push that in there. Okay, and we've got a little bubble of liquid on the top. That's alright because it's getting gradually drawn in by the flow cell. You can see it very slowly getting smaller. I'm not going to tip it up too much because otherwise I'll have buffer all over my laptop. Okay, and that's all been drawn in to the flow cell now, so it's been sucked up, and that's going to be about ready to take our DNA. So the next thing I need to do is take that buffer solution that we prepared earlier. I'm going to take it all up, mix it with my DNA, give it a really good mix. All right, and now we are good to go. So just want to make sure I've got no air in the bottom of it. That is much better. Okay. And we are going to put that right on the flow cell there. And we can just about see that little bubble of liquid there getting drawn in. Is it gone? It's almost gone. Right, so that's going to take a couple of seconds to draw in. And that's about it for now. So I am going to plug this in, switch cameras. I'll see you in a second. Okay, so I now have my MinION plugged into my laptop, and I just loaded up some software on the screen. So here we've got a couple of options. So I'm going to just click on my machine, and then I'm going to click on start. Oh, I'm not going to update right now. And click on start. Okay, so the sequencing run has started now, and the first thing that we have to do is warm the MinION sequencer up to 37 degrees, because again within the sequencer we're borrowing a lot of biological molecules, and these work optimally at kind of body temperature, so that's how we're going to run. So we've just got the MinION here, and that's just going to run for a couple more minutes. Okay, and here we have an example of some of the data that we can get from the MinION sequencer. So here, if we zoom in a bit, you can see all of the A's, C's, G's and T's that a regular file would look like when we create a DNA sequence. And this is just directly on my laptop, so we can use this to go ahead into various different types of analysis software, and we can start looking into all kinds of different questions. So that might be about rare diseases, or about heritability, or what makes different species unique, or about pathogens like bacteria and viruses. Basically, if it's alive, it's got DNA, and that would mean that we're interested in it.
Awesome. So that was the video that Kim very kindly recorded for us. I definitely think it's really interesting to see quite how small the sequencer is. So now we'll hand over to Leah, if you're sort of ready to take over from us, Leah. Awesome. I will let you sort of introduce and take over. And if you've got any questions for Leah as we go, please pop them in the Q&A or in the chat. And we'll either ask Leah as we go, or we'll save some for the end as well.
Sounds good to me. Also, just to say, if there's anyone got any questions about the stuff Kim was talking about, I might be able to get some of them, maybe not all of them, but I'll give it a go. Cool. Brilliant. So just to say, my name's Dr. Leah Chappell, and I'm a researcher at the Sanger Institute too, and I'm going to walk you through some different kinds of DNA sequencing technology. Hopefully I can share my screen so you can see what I see. And just cool. Do I have my title slide up?
Yeah, perfect.
Awesome. Cool. So I'm going to talk about different ways of sequencing DNA, including some ways of doing it from single cells. So Kim in her talk was talking about the idea of stealing some biological molecules to do useful things. Well, actually, all sorts of DNA technologies do this. So I'm guessing most of you folks who are watching have come across the idea that DNA is double-stranded before, which is kind of like this sort of left-hand side of the cartoon. And you've come across the idea that there are 4 bases in DNA, A, C, G, and T, and that there are base pairing rules to say that A can pair with T and that C can pair with G using hydrogen bonds. So I'm hoping that idea is kind of familiar to you, and we're going to build on some of that now. So that's how you get kind of one stable molecule. Actually, cells, as well as some of the stuff we steal the machinery for, copies that DNA. And the way they do that is that they unravel that DNA helix, kind of like, untwisting it. And they use each of these strands as templates to make more DNA. Now, there's an extra molecule here which is super important, which is the primer, which is a short, tiny piece of DNA or a similar molecule called RNA. And using the kind of base pairing rules that you get in helixes, that binds to bits of this single-stranded DNA. And with floating kind of free nucleotides, these A, C, Gs and Gs that are nearby, you can build 2 strands of new DNA. And if you look at the cartoon on the bottom, you can see how that is that each of the new double strands has one of the original pink strands and the second blue strand there. So yeah, that's what happens in nature. And basically we can hijack the idea that you have the free nucleotides, which are the building blocks of new DNA. We can hijack the primers that are used to get it going, and we can hijack the enzymes that are used to do this. And using those tools, we can do DNA sequencing. The other key idea, which I'll just show now with a couple of cartoons, is the building blocks for DNA, these nucleotides. So the base, which is like the bit that makes the A, C, T, and G different, is on one side, and there are other chemical parts of the chemical molecule, which we can talk through in the Q&A in a bit more detail. But what I just wanted to point out is that one point on this molecule, there are 2 sort of flavors. There's one that has just a hydrogen atom and one that has a hydroxyl group, or an oxygen and a hydrogen atom stuck together. Now, these have different chemical behaviors, and actually we can make real use of that to do DNA sequencing. And basically, although maybe it doesn't look a lot different here, that one difference between them can be used as a trick that basically underpins loads of genomics technology. So here's a kind of way to explain why they're useful. This one with the hydroxyl group only can't be extended into a chain. So when every time you incorporate that, the chain stops there. And if we've got kind of colored versions, you can kind of visualize that, right? If you incorporate that first, you just get a very short chain through to, if you happen to by chance incorporate it last, you get quite a long chain blocked at the ends. And basically you can get every combination there to read all of the letters. Now, in the older technologies, you do it one base at a time by running DNA through this thick, gloopy, well, they call it gel, it's like a slab gel, looks a bit like jelly, to be honest. And by separating, like using electrical charge to move the charged DNA through this, you can basically separate this DNA by size. And if you separate DNA by size, you can read off the sequence. So basically here we go, you got the red, the green, the yellow, all the way through. That's corresponding to red, green, yellow in the size. So that's a super neat trick, and it's how that was kind of developed in the 1970s. But, you know, like most old things, it's in black and white. So this is like an image of a really kind of old style Sanger sequencing gel, which is named after Fred Sanger, who invented this technology. And yeah, these are on big solid slabs of gels that are kind of like sort of A3 paper kind of size, big things that are hard to handle. And because it's black and white, you can only do one letter at a time. And although it's, you know, accurate, it is slow and you're limited. Basically, this is, you know, this technology was around the time of the Human Genome Project kicking off, but it sped up by becoming multicolor. So, you know, you tune in. So there's in the early 90s, you can add colors to those nucleotides that have the blocking group on. And if you have 4 different colors, you can actually read them all at the same time. And you get this trace like at the bottom. And basically having the 4 colors at the same time means you can just, you know, read along the DNA sequence by looking at the sequence of the color peaks. And this is neat because as well as being multicolor, you can run it in many samples at a time. This is meant to represent 96 samples moving along at once, and the colors being detected with a laser. So, you know, we've got a big kind of change in speed, moving from kind of black and white technology to like full color in parallel technology. And this is the kind of tech that powered the Human Genome Project. Now, I could talk lots more about the details of these, but I just want to kind of give you an overview of all the tech we have. One of the big changes kind of in the early 2000s, around when I sort of started working on these technologies, is the invention of so-called next-gen sequencing machines. And basically the idea of this whole kind of suite of new technologies was to read more DNA faster and cheaper. Now, there's a few different companies that made technology. The one I'm going to talk about today is from a company called Illumina, basically because that's the technology that turned out to be the most popular. That kind of won the day in the kind of race to be most popular. Now, here's some pictures of sequencing machines that are kind of like the ones you saw in the video clip at the very beginning. It's very hard to tell how big they are from this picture, but what I would say is that basically the kind of medium size, this one is roughly the size of kind of a washing machine at home, and this one is kind of almost as big as 2 washing machines stacked on top of each other, just to give you a vague idea. So very different than the size of the little sequencer that Kim was showing you earlier. Now, so these use Lite to read the sequence of DNA. So in some ways, and there are different colors depending on whether it's A, C, T, and G. So in some ways, they're very similar to the kind of capillary Sanger sequencing technology I showed you just before. But the neat trick here is how many bits of DNA it can read at once. Those other technologies were reading one kind of molecule at once. These, you're reading millions and billions of fragments all at the same time on something, a glass slide that's about half the size of a kind of debit or credit card. So I'll try and show you one of those at the end. Basically, we could talk lots and lots about the details, and I get super excited about this, but I'm just going to pick on a few key things to give you a chance of finishing on time. So, you know, this is not just a sort of static glass slide. It's actually full of these tiny kind of tunnels where fluid can go in and out. And as well as fluid going in and out, all the kind of enzymes and DNA building blocks can flow in and out as well. And then you can use that big sort of washing machine sized machine to take pictures of it. And you'll see that these spots. So this kind of looks like some spots I've seen on tiles on a kitchen floor, but actually in each of these spots, they've been made by like a single DNA molecule binding. So when you're looking at all these spots, that's how many different DNA molecules are binding in a tiny, tiny patch. The cluster density there, which is like how many spots there are, is showing this something like 1,000 per millimeter squared. And yeah, we've got many millimeters squared on this, so you can measure millions and millions of DNA molecules all at the same time. So I've got some cartoons to try and explain in a little bit more detail how this works. This is probably the most technical bit of the whole talk, so either pay attention or like come back to me in about a minute, so either works. But basically the bit of this technology is that you've got fragments of DNA, and by fragments, I mean rather than like the whole amount of DNA from a cell in one piece, I'm talking about chunks that are sort of 300 letters long. So 300 runs of 300 letters of runs of like A, C, G, and T in a double helix. So there's a couple of tricks that mean we can measure all these molecules at once. The first of which is that we add extra chunks of DNA of specific sequences to either end of those molecules. So they're called adapters by people who use the sequencing tech, but you could equally call them handles, right? The idea is that basically you don't know what these DNA fragments are, they're kind of random, but these handles that go on the two ends are all the same. And there's a couple of tricks which mean it's possible to make sure that each molecule gets 2 handles. In the cartoon, they're colored purple at one end and blue at the other. And the important part is that every molecule gets a different handle each end. Now, this is meant to be a cartoon of those glass slides I showed you earlier, that flow cell. And the neat trick about the flow cell is it's covered in some DNA molecules that are basically primers, like the ones I showed you from the beginning. So these primers are the same, complementary to the sequences stuck on the end of these. So using normal base pairing rules, you can use these to stick as handles to stick the DNA molecules to the glass slide, which is pretty cool, to be honest, that people have figured out how to do this using some neat chemistry. So once you've got your DNA molecules stuck to the slide, they tend to kind of bend over and kind of just flop around. Imagine flowers that you haven't watered, like falling over, or like kind of, and then that's roughly what's going on. Or you can kind of imagine it that, you know, they're sort of their feet are Velcro to the floor and they're bending over and trying to touch their toes. Like both things kind of work. And some of you may be familiar with some of this chemistry, some of it not, but I'll see if you recognize it if you've done some of these modules at school. But basically the idea is that you use these primers at the other end so that there's a sequence, and then you can use DNA replication enzymes like the ones in a real cell to help make a second strand of DNA. Then you can use a trick, like heat will work. You can also do it with some chemicals, and you split up the DNA double strand again. So you have two identical molecules. And you can basically do that again and again till there's a whole bunch of copies of DNA in exactly the same spot. Now, if that sounds a bit familiar to you, it's very similar chemistry to something called the PCR or polymerase chain reaction. So I'm happy to chat about that at the end if you're interested in kind of comparing them. But basically the trick is here that the DNA molecules are stuck down and that the copies are all identical. Then we can basically use some of the similar tricks before with these blocking DNA nucleotides, or the sort of building blocks. And the trick for the Illumina chemistry is that they've got fluorescent molecules in. So basically you can do some tricks that means you can have a stack of photos that correspond to the DNA sequence. And the trick here of getting this happen at once is that when you stack all these photos up, you can get different stacks of photos corresponding to like different DNA positions on the slide. So all of this stack of photos in the green box belongs to what was the green DNA molecule. All the stack of molecules in the pink box belong to the original pink DNA molecule, and all the kind of orangey reddy ones belong to that red molecule. Now, that's quite a lot to take in, and I'm happy to chat through it again at the end. But the things I just want to take away from that is that we can do lots of DNA molecules at the same time using fluorescent tagged building blocks, and that because it all happens on the flow cell in a very small space, you can do it with not too many enzymes. Right, okay, so that's a lot of technology. Again, happy to answer any questions in the Q&A about more of the details of that. I could talk about that for hours, and as Em and Fran could tell you, like we could do a whole talk on those nitty-gritty details. But I'm just going to kind of zoom out and give you a broader picture now. Now, what I was talking about is how many pieces of DNA there were there, and how many you can read. Now, that just makes so much data, it's kind of hard to get your head around how much data there really is. So here's a picture of some of the computer servers from the campus, and a bunch of these green lights represent hard drives stacked together really closely in these kind of double fridge size cabinets full of data, and these rooms are stacked full of them. So, it's hard to get exact numbers that are up to date today, but, biologists have generated so much data all over the planet for DNA that there's way more data than there are for like the big physics projects like CERN you might have heard of. And, we definitely, there's so much text data. It's great and exciting, but it involves a lot of computers to analyze them. And the thing that wasn't obvious to me when I was at school is how useful it would be to be able to write computer code to help understand these patterns of data. So if any of you are interested in kind of being a biologist later on, I would encourage you to try and have a look at like learning a bit of coding. Even a little bit is super helpful. Or if you're interested in coding already but don't really know what to do with it, maybe come and look at some biology data, because there's loads of new exciting data, and always more people are needed to analyze it and figure out what it means. So kind of Kim touched on this before. So like I showed you some washing machine size sequences. You can imagine they're not super portable. I would not want to take one in my rucksack, but the sequencer that Kim showed you, that's super portable. There are examples of people taking it to deserts, jungles, even out into space. So I think it's really cool that stuff is now sort of small enough to be portable enough to take anywhere. Now, there's, it's worth saying that, there's other sort of technologies are coming on the line. This is not the only company doing one. And I kind of speculate that one day You might have a DNA sequencer in every GP clinic so you can figure out what bug you've got. Maybe you'll get them in, to help figure out if food is past its best before dates. And I don't know, I might get told off for saying this, but I think, you can imagine one day where you have one in your kind of toilet in your house, so that if you can watch whether you're keeping a healthy diet by tracking the DNA in your poo and pee. So, you never know. Like, I reckon there's a future where portable DNA sequencers are just everywhere. So if that day comes, you'll have heard about it from us first. And just the last thing I want to say about the nanopore technology, but happy to chat more later, is that rather than taking pictures of DNA, like we were talking about before, this technology unwinds the DNA at double helix and runs it through a protein channel, and it uses electrical current to tell which base is which. So yeah, this kind of sequencing in the dark with no pictures. So that's just a very different way of looking at DNA than the other methods we saw before. I just want to briefly mention that we don't just have to sequence DNA. You may or may not have heard of a molecule called RNA. Basically, RNA is a molecule that allows you to copy out instructions from the DNA to turn it into protein. It's the molecule that goes in between DNA and making proteins that do something in the cell. And the thing about RNA is that different kinds of cells make different RNAs. So I kind of think of DNA as kind of a recipe book. And you can imagine that each different kind of cell uses different recipes from different pages. So these are kind of cartoons showing this, the idea that there's brain, liver, and lung. Like these are meant to represent genes, that some genes might be on everywhere. So these would be the boring genes that are kind of doing the same things, keeping the cell working. Whereas there'll be some genes that are only on your brain that are probably about connecting neurons together. There'll be some genes that are only on in liver, which will be about doing the functions of the liver, and there'll be some genes that are only on in lung. And by using the molecule we can basically do a couple of tricks so that we can use DNA sequencing machines to read RNA molecules, which is pretty cool, so that we can figure out which genes are on in different places by mashing them up. So that's like the technology that's existed for a few years, where we take a whole chunk of tissue and we mash it up. But now we want to do stuff from just single cells. So why and how, I guess, are the two questions. So why? So my kind of analogy for saying about single cell, so the kind of existing sequencing, is that you're basically mashing up everything that's there and turning it into a smoothie. And smoothies are great, and you can probably tell the difference between a smoothie that's mostly strawberry or smoothie that's mostly banana, but you can't tell how much exactly was in there. With single cell sequencing, if you measure one cell at a time without mashing them together, you can count how many strawberries, count how many pieces of orange, and have a much better understanding of how this works. So yeah, that's what single cell does. And basically, big projects are now kind of making a Google Maps of the human body by trying to take all the different cells from different tissues and take them apart one at a time and measure them one at a time to see what's there. And another way of thinking about it as well as a Google Maps, it's also kind of a bit like a kind of periodic table of all these different kinds of cells, from white cells in your immune system to brain cells to muscle cells, and to figure out how they kind of go together. So I just want to check on time. How long do we have left?
We've only got sort of 5 minutes left in total. So if anyone's got any questions, you can start popping them in the chat. And then what we'll do maybe is, Leah, if you want to sort of flick through your next few slides, but if any questions come in, I'll interrupt you and sort of ask those as we go, if that's all right.
Yeah, cool. I thought I'll just pick on one of these. That's the coolest one. So basically, another way to do this, we can isolate individual cells in drops. So here's a picture I took in our lab, and these are individual tiny droplets. And basically, the things that the red arrows are pointing to are individual cells. And what we can do is encapsulate individual cells in droplets. I'll just skip forward a couple of pictures here. the neat thing about being able to encapsulate individual cells in droplets is that we're then able to isolate them, measure them, and try and understand how that means. So basically, I work on the technology that does that. And one of the ways that we do that, as well as droplets, are these devices that can fit onto a microscope slide. Now, with this picture, it's really, really hard to tell how big that is, but it's actually on something about half the size of a debit card. And it's not super obvious in the picture, but each of these squares here corresponds to like 150 individual squares. So on the whole thing, it's like 100,000 wells, which means we can measure loads and loads of cells individually by letting them drop into these wells where they can fit. So I'll show you one of those in a second and we can chat about it. But yeah, basically, these allow you to measure one cell at a time, and then we can figure out how to put the cells together, how the cells go together in this periodic table to make the tissues. So I think I'm going to stop there for a sec and yeah, take any questions you've got.
I think we've had one group that have just left as their lunch time has ended. So we've got one other group, if anyone from that group's got any questions. Otherwise, if you think of any questions after you finish the session, you can always email us and we can ask Leah or Kim for any sort of extra answers.
So just while folks are asking kind of questions, here's that device I was just talking about now, the microscope slide, and here's how it can kind of compare to a normal bank slide. So they're neat, these things, like how, like, I guess it's easy to forget how really tiny individual cells are. But yeah, we made these ourselves from stuff we have on campus.
Great. While we see if any questions come through in the Q&A and or the chat box, I'll do my usual, Leah, and ask you, so how did you get into the job that you're doing now, and sort of what was your career path getting to where you are?
So I guess, I was always really keen on kind of watching nature documentaries and trying to understand the natural world, and I'd picked biology at school, and I hadn't really, but I'd also picked other subjects like geography and chemistry and hadn't really decided what I wanted to do. But around that time, there was the 50th anniversary of the discovery of the DNA double helix. So I remember sort of seeing TV shows and reading magazines about the DNA double helix and thinking, oh, DNA, that's cool. So I ended up doing a degree, which was biochemistry mostly. So that's, yeah, all about enzymes and trying to understand DNA sequences, basically, my degree. Then I actually came to Sanger as a PhD student. So that's what I did after I did my 4 years degree after school. Sort of started learning the technology there, and I just got sucked into playing with this DNA technology. Like, I really love all this sequencing tech. I actually ended up using it to study malaria parasites for a while, but now I mostly work on kind of the pure tech development, which can be used for a whole bunch of different things. But yeah, getting distracted by wildlife TV documentaries and reading some science magazines, and then doing biology at university.
Fantastic, brilliant. So there isn't anything that I can see on the chat. Can you?
Yeah, I don't think we've got any questions coming in. So I guess that brings, I mean, perfectly on time. That brings us to the end of our session today. As I said, if anyone thinks of any questions after today, feel free to email us. We can ask either Leah or Kim for any extra answers if you think of anything else. But thank you very much, everyone, for joining us. Thank you very much, Leah, for taking time out of your day to come and talk to us as well. The sign-ups for next week's sessions, which actually we've got a few scientists from Illumina Lab joining us. So when Leah was talking about the Illumina sequencing, we've actually got some scientists from that lab coming in to talk to us about their career pathway. So if you're interested in that, sign-ups are on our website, the same place as you registered for today's session as well. So hopefully we might see you there. But yeah, thank you very much, everyone, and have a nice rest of the day.